It's going to be what we call lightning talks, or kind of quick, fast-paced presentations. Each of the 6 speakers gets about 5 minutes, so 3 to 4 minutes for their presentation and about a minute for Q&A. It's designed to kind of give you a quick run-through of the projects they're working on and hopefully spur you on to speak to them more about their projects in the networking after this is over. So with that, the first presenter is Aiden Cicchetti. How you doing? He's got the timer right here.
Okay, cool. Hello, everyone. So my name is Aiden Cicchetti. I'm a PhD candidate working with David Sedlak at UC Berkeley, and I'm studying nutrient removal in a new type of constructed wetland called the horizontal levee. So there are 3 major problems that are faced by urbanized estuaries like the San Francisco Bay Area. First is the release of contaminants from wastewater treatment plants. Second is the loss of historical wetland habitat. For example, San Francisco Bay has lost 95% of its wetlands in the last 2 centuries. And the third is that urbanized estuaries are at a heightened risk of the effects of climate change, such as sea level rise. So traditionally, flood control for coastal areas would be achieved using levees that are armored with riprap and have little to no habitat value. We've been studying a multi-benefit alternative called the horizontal levee. These systems are used to polish wastewater that's fed through their subsurface while restoring wetland habitat and also protecting levees. So to test the water quality aspects of these new systems, a field site was constructed at Oro Loma Sanitary District in San Lorenzo, and it receives nitrified effluent from the wastewater treatment plant that's pumped into the subsurface, and the wetlands divided into 12 wetland cells that allow us to test different design parameters, like soil type. So traditionally in engineering research, treatment systems would be studied by tracking influent and effluent contaminant concentrations, but this doesn't give you much information about what's happening internally. So we've tried to approach monitoring more holistically. And to do this, we've tried to characterize internal spatial variability. We've been sampling multiple compartments like plants and soils to try to understand removal mechanisms. And we've also tried to understand how other benefits like habitat quality and climate change resiliency relate to these. So what we found with this approach is that a large part of this internal variability in water quality is related directly to hydrology. We've characterized hydrology by tracking certain water chemistry parameters like conductivity to calculate evapotranspiration rates and nitrate concentrations, which we've used to calculate the relative internal flow rates, like overland flow and subsurface flow. We found significant subsurface removal of contaminants using pore water sampling, and also we've shown that there's a strong link between contaminant removal and subsurface flow rates. Additionally, when we eliminate overland flow, as shown here, we observe greater than 92% removal of nitrogen species, pharmaceuticals, and also pathogen indicators, compared to 20 to 80% removal that we normally would see. So the key takeaway there is that hydrology is controlling contaminant removal. An example of the multi-compartment sampling we've been doing is how we've been tracking nitrogen isotopes to understand nitrogen cycling. And so nitrogen comes in as nitrate in the influent wastewater, but there's also a large reservoir of nitrogen in the soil. And these 2 sources of nitrogen have specific isotopic signatures that we can think of like fingerprints. And so soil nitrogen isotope signatures tend to dominate in subsurface systems, but as wastewater is introduced, that isotope signature gets incorporated as well. And plants can take up nitrogen from both these sources, and so the isotope signature of plants ends up reflecting the relative contribution of those 2 sources. Using this understanding, we've been tracking uptake of wastewater nitrogen by tracking plant isotope signatures. The chart to the right, which is horribly misshapen now, plots the nitrogen isotope signatures of plants over time, and there are 2 boxes which in the past showed what the wastewater isotope signatures and soil isotope signatures were as well. And what this chart was supposed to show is that the signature of plants has slowly shifted to match what we see in the wastewater. And so when we pair this with the bulk biomass measurements, we've estimated that 30 to 50% of applied nitrogen was taken up into plants in 2017. But this leaves about 40 to 60% of removal unexplained. And that's likely due to a combination of various microbial processes in the subsurface, but we're going to use additional isotope tracer methods to quantify those. So the final part of our approach is this monitoring of additional benefits, such as habitat quality and also climate change resiliency questions. And a lot of these questions relate directly to the amount and quality of biomass. And in our site, we've seen a significant growth of biomass, but we've had to partner with other people to do some of these measurements. For example, we partnered with Save the Bay, who did plant monitoring to reveal that less than 2% of the plants at the site are non-natives. And also we've been partnering with Planet, who actually is here, and the Alameda County Mosquito Abatement District to calculate biomass growth rates, which we know control accretion rates and wave dissipation potential. And so our results have important implications not only because horizontal levees are being constructed around the San Francisco Bay Area to try to combat these issues, but also because we've shown that they can be effective in polishing wastewater, and we've developed some new systems for studying them more holistically. Thank you.
Oh, actually, we do have a minute for questions.
Oh,
I
okay.
forgot about that. Sorry.
Yeah, so, and I can answer questions.
Thanks, Aiden. Next up, I have Wylie Jennings, and followed up by, it's not in exact order, so I'm gonna get you prepared. The next is gonna be Emanuel Rodriguez or Sadie Gill, followed by Victoria McGruer and Wynn Cowger, followed up finally by Matt Heberger. Thanks.
All right, thanks for hanging in there, everyone. My name is Wylie Jennings. I'm a PhD student at Stanford, and my research group generally studies fecal indicator organisms, microbial pollution at beaches. So I'm going to be talking about citizen science water quality monitoring project we've been running the last couple of years, looking at a couple of freshwater discharges at beaches in the area. So recreational waterborne illness remains a significant health problem actually in the United States. Rivers and creeks, especially in urban areas, are common sources of fecal indicator organisms to beaches. And because of this, some jurisdictions, such as San Mateo County, actually provide guidance to beachgoers, such as remain 300 feet from a flowing creek. However, the water quality monitoring that's typically conducted at beaches can't actually determine the spatial extent of pollution from these discharges. So the goal of this project was to try to take a couple of popular recreational beaches in the area, do some sampling, and see if we can try to estimate what safe distances from these discharges actually look like. So to do this, we used simple salinity measurements as a conservative tracer of freshwater flow from these discharges in the surf zone, just to try to put spatial bounds on the freshwater plume. We recruited 50 volunteers, and we had about 40 people actually collecting samples. And we asked them to go out once per week for 12 weeks, spanning the wet and dry seasons, each time making at least 5 measurements along the shoreline. So we ran this project in 2017 and 2019, but I'm going to be focusing on data from 2019. And the reason we want people to make at least 5 measurements each time they go out is because we want to estimate the distance along the shoreline at which freshwater is diluted by 90% in the marine water, because typically diluting the fecal indicator levels in these discharges by 90% would result in water considered safe for swimming. Our volunteers also dropped off samples for us so we could validate 20% of the measurements made. And these are the data collected by our volunteers. We had 1,452 measurements submitted, and using very strict quality control criteria, we were able to use 650 of these measurements to estimate our 90% dilution distances. On the left, I'm showing data from San Pedro Creek in Pacifica, and on the right, San Lorenzo River in Santa Cruz. So San Lorenzo River often has about 100 times the flow of San Pedro Creek. So the yellower data points are fresher water, the purpler are more saline water, and you can see that change varying from the mouth of those discharges. It's important to note here that these are the data aggregated. When we're actually estimating this dilution distance, we're treating each profile individually. Okay, so here are those 90% dilution distances. On the left, we have San Pedro Creek, sorry, it's labeled as Lindamar here, and on the right, we have San Lorenzo River. The basic takeaway here is that at San Pedro Creek, the median 90% dilution distance is about 125 meters here, which is not too much bigger than the 90 meters the current guidance provided by San Mateo County. At San Lorenzo, the distance is around 400 meters, although distances in the 600 to 700 meter range are not uncommon. Okay, so that's kind of the physical science aspect of this project, but I also want to get at volunteer participation in this project. So because encouraging sustained participation in a project like this is super crucial to their success, we wanted to explore what kinds of project management, project design parameters we could maybe tweak and see how that affected participation in the project. So in 2017, we ran this project basically with very low communication with our volunteers aside from an initial training. And in 2019, we had a lot more communication with our volunteers. These are different groups of volunteers in each year. So in 2019, we also had twice monthly emails with our volunteers, sending them general project updates, ways to improve the data collection process. And what you can see overall is that we found no effect on sampling effort from having a much higher communication with our volunteers than having lower communication with our volunteers over the 3-month timeline of this project in each of those years. So in the 2019 iteration of this project, we wanted to take this question a step further because through surveys in the first iteration of this project, we found out that many people who participated were motivated to participate because they wanted to learn about environmental or scientific issues at their local beaches. So we wanted to know whether providing increased scientific feedback to some of our volunteers would increase sustained participation in the project over those 12 weeks. So we randomly divided our participants in half, and all participants received the same twice monthly general project updates, but half also received more intensive scientific feedback about the data that had been collected in the past couple weeks, as well as relevant fecal indicator levels and flow data from those discharges. And again, what we find is overall no detectable effect on sampling effort from people who are receiving increased scientific feedback. So the coloring here represents the number of these data reports that people were actually looking at. And you can see that people who were in the lighter colors on the right are actually the people who are looking at more reports are also sampling more frequently, so that makes sense. They're highly engaged with the project. But there's also a group of people who don't have access to this increased scientific feedback who are sampling frequently as well. So overall, there was no effect. I'd really love to talk to people more who are working with community monitoring about engagement in these products or projects, quality assurance measures, and water quality with communities in general. So thank funding, NSF, other students who have worked with me on these projects, local organizations, and most of all the volunteers who collected data for this project. Thank you very much.
Lots of questions today. Thank you. So the slide that you had that mapped out the number of reports viewed that had the coloration on it, and then you said that there wasn't an impact. I mean, can you help me? Because I'm seeing that, and it looks like the more you communicate with the volunteers, they actually read more reports than the group that received less communication. So it looks like the opposite conclusion, unless, like I say, I'm interpreting that wrong. So
yeah, so this plot is on the left. So we asked people essentially to go sample 12 times on 12 different days, and then half of those people got these data reports. So those are the people in the yes category, right? So overall, if you just compare the two columns, the yes and the no, if one of them were significantly shifted higher than the other, then you could say, look, it looks like there's an effect of receiving this treatment, but we don't see that. Overall, basically kind of the typical number of days that people are going and sampling is quite similar between the two treatments. Does that make sense?
yeah, it just looks like they're engaging more with the feedback on the right side, and I think maybe I'm just like misreading it somehow.
If you look at the bars in the middle, which is kind of the median,
Okay.
they're quite similar.
Okay.
So.
Okay.
You mentioned the quality assurance. So it went from 1,400 to 600.
Right. So some of these measurements were discarded for the analysis due to kind of typical quality assurance concerns like accuracy. Some of the kind of GPS coordinates were not valid. But some of them also, a good deal of them actually were discarded because they weren't useful for making this specific analysis. So it is possible, for example, if we wanted to estimate a distance along the beach at which freshwater was diluted by 50% instead of 90%, which perhaps is less health relevant or less intuitive, then we would be able to include actually a good deal more of those data points. So the way I frame this, and we could talk about this later, is I have a whole flow chart of quality assurance measures, and you can go through and kind of treat this almost as a sensitivity analysis and tweak each one and see how that affects your results. Yeah.
Sadie Gill or Emanuel Rodriguez?
Hello, I'm Emanuel Rodriguez. I am a data scientist at a consulting firm called Flow West. We're based out of Oakland, California. And today I'm going to just talk about how we use R to help us package data and make it more accessible. So we all know that R is really good for data wrangling, doing exploration visualizations. But what sometimes we don't know is that we can package our data into R using whatever binary formats we need. And if we ever embarked in like a wrangling task, we know that there's a ton of decision making that goes on when we get the raw data to when we deliver the data. So when we package this up in an R package, we're able to capture all the processing tasks that we want. And with this, we can build a lot of the testing suites that goes around with when we develop an R package. We can build on top of this if we want to source or host our source code on GitHub. So this way we can not only track like our input into the source code, but also track our collaborators and however we want to share the code. Second thing we want that we get from building packages, data packages with R is improving accessibility. So this is an example of a package we've developed at Flow West called CDEC Retrieve. We kind of wanted to emulate a lot of the work that USGS did with the data retrieval package, as well as what I think Berkeley Labs put together an R-NOAA package for the NOAA APIs. So we did the same thing. We did the same thing with CDEC data. I don't know if everyone's used CDEC, but it's kind of like a California-only sort of USGS kind of thing. So being able to use all these three packages, we can build really quick. We can prototype these dashboards super quick. So in the bottom left or bottom right, you see one of our dashboards where we're just porting real-time data that uses our CDEC package as the backend. And sort of the last thing we want that packages enable to do is properly document all of your source code and your data as well. So in this case, this is actually a package that was put together by my colleague, Sadie Gill. This is package that was developed for the CVPIA SIT team, who uses a lot of the calcium data input into their fish models. So what she did was do a lot of synthesis work on the calcium data, produce the new data, and document all of the work. Kind of the big thing to take away from this is that there's this package called package down from within R that enables you to take your formatted code, your formatted source documentation and make a super snappy website out of it. So actually what you're seeing on the right over there is just a screenshot of the website, and she details every single thing that you need to know about the data. And that's it. Thank you. Are there any questions?
I'll introduce myself. Hi, everyone. My name is Tori. I'm a PhD student at University of California, Riverside. I'm going to take things in a bit of a different direction and talk about toxicology and how we're using these larger data sets we're accumulating to understand patterns of toxicity, and in our case, how crude oil impacts fish and fish development. So since 1969, there have been over 40 major oil spills in the U.S. These range from Deepwater Horizon, the largest marine spill, to those in freshwater systems, such as the Kalamazoo River with a pipeline burst. And so with an oil spill or when we're planning for an oil spill, we're interested in kind of what are the ecosystem impacts and how are the resources we utilize impaired, right? And so we're fairly good at understanding acute exposure, so maybe what concentrations cause lethality in fish. But we're not quite there yet with modeling and predicting sublethal effects. So at what concentrations, sometimes parts per billion, do we see differences in embryonic development in fish, is what I'm going to focus on here. So one of the most sensitive organs in fish development with oil exposure is to the heart. We see abnormalities in heart development across many species with exposure to crude oil. And so with this, on the organism level, we see reductions in swimming performance later in life, even if they haven't been exposed to oil since those first few days or weeks of development. And so with this, we are concerned with population level impacts and reduced fitness over that whole population. But what we don't understand here is how oil, or phenanthrene, the key cardiotoxic component of oil, interacts with molecules in the larvae to produce this toxicity pathway we see, so what we call the molecular initiating event. And so to make predictions about what this mechanism might be, we turn to several recent transcriptomics studies. So these are studies which compare gene expression in either control or oil-exposed larvae. And what I want to highlight here is that again and again across studies, across species, we see cholesterol synthesis as one of the top impacted pathways. So this is something we really wouldn't have focused on if we weren't looking kind of more broadly across these exposures in these studies. And so I'm going to talk a little bit about cholesterol in the heart. And what we think about when we think of cholesterol in the heart is not necessarily good, so high cholesterol, not good for your heart, right? But what we think is happening here is that crude oil or phenanthrene is actually reducing cholesterol in the heart of these larvae, or in these larvae, and that is impairing heart function through changes in cell structures. And so with this kind of a really quick overview of what we've been doing in the lab is, so on the left here we have our control larvae, and on the right we have our phenanthrene exposed larvae, so a proxy for crude oil. And then on the bottom two, we're staining for cholesterol, and we see that, so this arrow is pointing at the heart, and there's a reduction in staining in that area, whereas we have specific staining for cholesterol in the heart of our control fish. And this is paired with a reduction in heart rate. So we have more work to follow up on kind of the connection of these two and whether this is really impacting heart function. But what I want to highlight is that understanding the molecular initiating event, or this mechanism, can help us make predictions in species we cannot test and species-specific sensitivity. Also, so kind of more broadly, omics technologies such as transcriptomics can help us recognize patterns across species when we're looking and trying to come up with hypotheses about what these mechanisms are, so help us derive to unbiased research questions. I had no idea I was going to study cholesterol in fish, but here we are. And transcriptomics and kind of looking at these broader data sets has helped us focus our research questions in this way. But there are challenges to this, so wading through kind of what is a general stress response versus what is specific to the question we're asking. So we have these larger data sets we're looking at. How do we focus in our questions and kind of wade through that as well? And that will come with kind of doing more of these studies. So I'd like to thank our funding sources and my lab and the team behind this, and I will take any questions.
Cowger. How you doing, Win?
Hi, I'm Win. I'm a PhD student at UC Riverside. I study trash, and I try to identify the sources of trash. We have a really cool datathon that happened today, all about trash data, so I'm a trash data geek. And so I'm going to talk a little bit about my motivation. These are the images that keep me up at night. This is what gives me my oomph. So here on the left, if you had to take a guess, where would you think that these images were taken? This isn't China. It's not India. This is Southern California. These are the streams in Southern California. No, these are in Corona, but pretty close. What's really bad about this amount of trash that's in the streams, and what's challenging about it, is that it doesn't stay in one place. So this video shows trash flowing down streams during the storms. It's really hard to manage things that are moving around. We can't just go and clean it all up. I sample trash moving through these streams to try to figure out how fast it's flowing through. And after a 2-minute sample with this net, I found all of this trash. It's like a handful of trash in 2 minutes and through this small net. So it's a really big deal. And this is really challenging and scary because we know that these plastic particles can cause harm to animals. Large trash can cause harm to infrastructure. And it is super expensive to manage trash. So I don't know if you knew this or not, but hundreds of millions of dollars are being spent every single year to manage trash in California. That's a huge number. And with that, we have a great opportunity to collect really detailed information to figure out what the best management strategies are, right? So my question is, where is the data? And also, the Ocean Protection Council doesn't know where the data is either. This is a recent report that basically says, like, we wish that we had some data on trash, but the best thing we have is some volunteers who have collected data on beach trash. I've looked on CDIN and the California Open Data Portal. You will not find any trash data there either. So this is a big deal. We need some information, at least a little bit of data on CDIN and the California Open Data Portal. And I'm super passionate in 2 years, we're going to make that happen. We're going to see it. And this is how we're going to do it. So I'm involved with 2 different groups right now. And if you know of trash data, or if you have trash data, I want to talk to you. I want to work with you to figure out how we can get it out there. I want to figure out what kind of visualizations would be useful for you, for your management strategies. We have 2 different teams that we're on right now. One's with the Ocean Protection Council. The other one's with the California Water Monitoring Council Trash Group to do this. We have a great team. And I'm so passionate about this. I've created a open access database so that if you are looking for trash data, you can find it. Just take a photo of this Google Drive. If for some reason you can't make bit.lys out of a Google Drive link, I don't know why, it just doesn't work. But take a photo of that or come see me after and you can get some trash data. All the data that I've compiled so far is there. It's going to be on California Open Data Portal in the next 2 years for sure. But it's not there yet. And to give you an idea of some of the cool things that we're doing with this data and what we were talking about at the Datathon, this is a little bit blurry, but basically this is a framework for, you have all these people that are collecting data on trash using different survey sheets. They have different names for everything. And so we've created a framework for merging all of those different names. And that's what that is. And we worked on that a little bit at the Datathon too. So we're getting better and we're going to be able to make these data sets speak to each other really easily once we have them all in one place. So please reach out to me. My contact information is there on the screen too. Thank you.
Any questions for Wynn?
Aren't there trash TMDLs and dump the regional boards? I would think so too. Isn't there data? There are trash TMDLs, and there should be data that is being created because of them. There are a few TMDL reports which have data. The biggest problem is that there is no there's no regulation for how you should report your data. There's no standardized framework for even collecting data. So, I mean, most of the TMDLs and the propositions that I'm seeing right now don't really have data.
Thanks, Wynn.
Thanks, Wayne.
If you were here.
Is there a mouse I can use?
There's not.
Oh, there's not?
No.
Can you drive?
Stand over here.
Okay. I'm going to give my presentation from over here. All right. Well, thanks everyone for sticking around. I'm Matt Heberger from the San Francisco Estuary Institute, and I'm standing over here because I don't have any slides. I just wanted to show you a little web app that we developed, I guess about a year ago. And this was, don't need PowerPoint. Let's see. The purpose was, so I manage the Delta Regional Monitoring Program, and the Delta RMP is, it's a program to measure a bunch of different water quality constituents, and one of them is pesticides. And it's a real stakeholder-driven program. We've got 50 different entities that pay into the program and fund us, our work, and keep close tabs on our work. And we're not so interested in producing yet another report, written report, but providing data out to the stakeholders so they can look at it themselves, kind of draw some of their own conclusions. And so we've got measurements of 174 now. For the first couple of years, it was 152 different chemicals, and we were measuring them at 5 locations, which represented inflows into the delta. We've subsequently totally changed our monitoring design, and now we're doing a more of a random trial at all kinds of random locations. So measuring at these 5 locations, inflows into the delta, 152 chemicals. So if I told you that there were 2.3 nanograms per liter of atrazine in a water sample, like what would that mean to you? Next to nothing. I mean, just a concentration doesn't really tell you much. And one of the ways we try to turn this data into information is comparing it to ecotoxicological thresholds. So could that concentration cause harm to fish or to plants or to invertebrates or things like that? And so this is a really simple data visualization. I call it a visualization, not just an infographic, because the key is it's interactive. You can interact with it, you can change things, you can filter. So here are the different chemicals. You can look at different locations, and let me make this bigger. So this is the kind of the cool, oh boy, it's not working well today. You can only see one chart. There are other charts as well. Maybe if I try to reload. Let's try this one more time. Huh, okay, well, I didn't test it in this browser, obviously, before I came. But if you look at sort of some of the different chemicals to see how they're doing, atrazine, azoxystrobin, try to look for one that has some more interesting data. So imidacloprid, if I'm pronouncing that right. There's currently a TMDL under development in the Valley for this. And so these lines here represent aquatic life benchmarks. So that's a concentration at which you might expect some kind of negative impact to fish or wildlife. And so you can see the number of exceedances where it exceeded the concentration, and you can turn some of these off, say, you don't care about fish, you're really only interested in invertebrates, you don't care about plants. And so you can just sort of see, looking at this line, like how they compare. And I guess in other browsers, you can see there's some different kinds of charts as well. And this was all made just in HTML, JavaScript. We didn't use any kind of fancy libraries. And I guess I kind of want to give a plug to using sort of free open source software, because it's sort of sustainable software development. You can find a lot of developers that know JavaScript. It's a really common skill set. It's like a fundamental building block of the web. I don't think you can find as many developers that know some of the more specialized software that we like to use sort of in our domain. So that's it. It's just a really simple kind of data visualization with a really specific purpose to look at are pesticides potentially causing harm to fish and wildlife in the Delta. Thanks.
Any questions for Matt?
You can, if you scroll down, so you've got your options up here. You've got some graphical results, and if you go down here, there's a little data table that you can copy and paste them into a spreadsheet. That's a pretty simple, silly way to do it. All this data is also up in CDN and NWIS and all the different online databases as well.
So it was really easy to wrangle all those different data sources and make them work together?
Well, this is
NWIS
just our program's data. So this is a really small, simple, I mean, I can see an area to expand this would be to, get all of the pesticide data in all of CDN and put it into here. Another way would be, there's additive toxicity is a thing. Instead of just looking at chemicals one at a time, having some kind of an index of toxicity in here. There's a lot of things we could do, but this was just meant to be like simple, quick, answer some really specific questions for a really specific audience.